Hey everybody! Today we're going to be talking about the blood agar medium. And the blood agar medium is a complex medium that is generally used to grow fastidious bacteria or bacteria that require complex or particular nutritional requirements. And that's kind of one reason for this type of plate. Another thing that we can use it for is to look at a different type of hemolysis or hemolysis, however you want to say it. And that's commonly used to classify different types of Streptococcus species, especially some of the more pathogenic strains. Now, with the intro out of the way, let's talk a little bit about the media basics. Our medium is basically just a standard growth medium, and then after you've autoclaved it and sterilized it, you then have to cool it down and you add 5% blood to the medium, hence the name blood agar. Now, normally this is like synthetic sheep's blood, or it can be real sheep's blood, just kind of depends on who makes it. But different bacteria will have different types of toxins that have hemolytic abilities, or the ability to break down red blood cells. So with that in mind, we can end up getting three types of hemolysis on our plate. We can have gamma hemolysis, which is no hemolysis. We can have alpha hemolysis, which is partial destruction of our red blood cells. And then we can have beta hemolysis, which is complete destruction of our red blood cells. Normally, gamma hemolysis will just look kind of like normal growth on a plate. Alpha hemolysis will look really just kind of like small clearances around our plates, maybe a bit lighter coloration on the plate. And then beta hemolysis will either look like a complete clearing around our colonies, or it will look kind of greenish. So with our media basics out of the way, let's look into or move into our results and kind of talk about how to score our plates. So we'll kind of start with the easy one. And I guess basically as we move through these, we're going to talk about, again, the different levels of destruction that we see. And basically, the clearer your plate looks, the more you're moving from a gamma into an alpha into a beta hemolysis. So let's start out looking at what gamma hemolysis actually looks like. So here's just two examples of what that could look like. Again, this is just our normal plate with some growth on it, and there's no destruction of those red blood cells on it. Now, if we move into something like our alpha hemolysis, this is where things can get kind of wonky. We can have weak ones and strong ones, and because of that, we can get varying degrees of our partial destruction here. So if we look at something like our Pseudomonas here, when we're looking at it from this view here, it doesn't really look like he's done a whole lot, maybe a little bit along the edges. But when we then look at it on the back, put the lid back on here, you can kind of see how he shows up a bit more than some of the other ones. So in this case here, that would be kind of a very weak alpha hemolysis. Now to look at one that's a little bit more straightforward to see, we have something like this right here. This is, again, alpha hemolysis. And in this case here, we have, again, kind of that clearing of the media, but we're not seeing a defined complete destruction along the edge of the colonies. So those would be examples of alpha hemolysis. Alpha hemolysis is going to be probably the most common type that we will see in our lab. Now, in terms of gamma hemolysis, beta hemolysis, in terms of beta hemolysis, again, we can kind of see it in two ways. We can have something like this, where we get that very, very green color around our colonies, and that's a good example of a beta hemolysis. We can also get complete destruction, and that's kind of what we're looking at here with this guy up here. If you're looking along the edge, you can see how that all of that red color is gone, and you can even kind of see my gloves through it. Another way to really test this one out, that's kind of an easy way to test whether or not you have alpha or beta hemolysis, is by taking some notes, and then if you take your plate up to it and you can see the letters through the clearing, then it's going to be beta hemolysis. Now, kind of with the results out of the way, in terms of inoculation, really the way that we're going to inoculate these is probably just going to be from just a broth to plate transfer. That's kind of what we do most commonly. If you have your sample on a slant or on a plate, you can do a slant-to-plate or a plate-to-plate transfer. For some of our labs, we might even do swabs, where we'll do either like a throat swab or a nasal swab, and we will then go ahead and inoculate them with those swabs. It really just depends on what lab it is that we're doing. But that should cover what we need to know for the blood agar plate.